Следующий доклад представит наш коллега, наш коллаборатор из Индийского института науки Mohit Jolly. Я сначала скажу на русском. Доклад будет посвящен механизмам и последствиям гетерогенности опухоли с акцентом на уровень единичных клеток. The next presentation will be presented by Mohit Kumar Jolly from Indian Institute of Science. Mohit will tell about mechanisms and implications of single-cell heterogeneity in tumor. Mohit, please.
Can you hear me?
Yeah.
Okay, great. I'll just share my screen. All right, I'll begin with thanking Evgeny with this kind opportunity to present some of our work at this exciting meeting. What I'll be talking about today is the implications and mechanisms that underlie these ideas of single-cell heterogeneity, which clearly various other speakers are investigating using sophisticated methods of single-cell RNA sequencing. So our lab doesn't focus per se on conducting experiments at single cell level, but we do analyze the data and from a mechanistic perspective, and then try to delve deeper into the implications of having this kind of heterogeneity in a tumor microenvironment. So mostly our work focuses on in the context of metastasis, which as you all may know, still remains the cause of 90% of all cancer-related deaths, where cancer cells spread from one organ to another using this fantastic set of freeways in our body called the blood vessels. And of course, it metastasizes via lymph vessels, and more recently, some literature via nerves as well. But blood-borne dissemination happens to be the most dominant one as reported in the literature. Now, if we look at the dynamics of metastasis, it's actually a highly inefficient process. It's a very, very small, minute percent of cells that leave the primary tumor that are eventually able to form metastasis. So our group tries to understand what are the special properties of these cells that are able to successfully navigate all the different bottlenecks that these cells are likely to face. Now, if you think more about the process of metastasis, as I was saying, it's a very dynamic process where cells continuously need to change their properties on multiple axes simultaneously, be it their ability to adhere to their immediate neighbors. So they have to weaken their bonds with cells in the primary tumor and eventually able to get launched in the bloodstream. And then once they reach a distant organ, they again have to reform those bonds and grow a whole new tumor. Of course, the ability to migrate and invade, the ability to evade attacks which are being done by the immune system as well as by different therapies that are being given. And of course, settling down in an organ which is completely different at biochemical as well as biophysical levels. So how are cells able to alter these different properties simultaneously is what we try to understand by developing mathematical models of regulatory networks involved in one or more of these axes and see how they coordinate those different properties together. Now, for metastasis, what has not yet been identified is a unique genomic signature, unlike for primary tumor, where we have collected extensive amount of data in terms of being able to collect different clonal evolution dynamics and so on. In the context of metastasis, we still don't have a unique signature that can be associated with metastasis. This might sound counterintuitive to many of you, but to me, this makes perfect sense because during metastasis, when the environment of the cell is continuously changing and presumably the variability in the environment is more than what is there in the context for primary tumor, what they would most likely benefit from is their ability to adapt rather than their ability to continue to adapt rather than their ability to adapt just once in an irreversible sort of hardwired mutation manner per se. And of course, then there can be advantages of cells moving together, which has been reported in the literature that when cells move together, they form more metastasis. Why they do that is still being investigated in the field. What I'm going to focus on today is this idea of non-genetic heterogeneity instead of the very well-established paradigm of genetic heterogeneity in the tumors. So non-genetic, as the word suggests, indicates properties which are usually not seen in genetic heterogeneity or genetic evolution, which is that the non-genetic components may not be heritable. There is some possibility that they may be inherited depending on what exact component is altered. Genetic is, since it's irreversible, non-genetic is reversible. Genetic is again deterministic. Once you have it, then all the next progenies would have it. Non-genetic is stochastic. There is an element of probability that is associated with it. This idea of non-genetic heterogeneity or phenotypic heterogeneity is relatively new in cancer, but it actually forms the central basis in developmental biology. So imagine a progenitor cell which is capable of giving rise to two or more cell types, in this case, two cell types shown here. So what decides which cell fate it gives rise to? There is an element of stochasticity that is involved in it, which depends on various factors, and of course in the cellular reprogramming. In the cancer literature, this is becoming, as I said, more and more prominent more recently. And of course, with the advent of high throughput technologies such as single-cell RNA-seq, which is this one of the major focuses of this workshop, we can now collect data and analyze and identify different tumor subtypes based on the proportion of different cells seen in the tumor and or in the microenvironment. The questions that we focus on sort of take on from here and start to delve into where exactly this non-genetic or this phenotypic heterogeneity is coming from, what is the role of this heterogeneity, functional implications of this heterogeneity in metastasis as well as in drug resistance. And given that cancer is sort of constantly evolving, can we find ways to control this heterogeneity? Because clearly this heterogeneity has been mentioned as a roadblock in terms of effective treatments, and some of which I will also briefly touch upon. So most of our work focuses on this transition, which is very well studied in the context of metastasis, which is epithelial to mesenchymal transition. The idea being that if you think about carcinomas, which are epithelial cancers, those cells usually do not have the ability to start moving. They are actually adhering to their neighbors, and they undergo this transition to be able to gain some of those properties. And these properties of migration and invasion usually being characterized with a mesenchymal phenotype in the body. So this transition is known as EMT, or epithelial to mesenchymal transition, which usually forms the first step of metastatic cascade. And once they reach a distant organ, these disseminating cells are presumably sort of sacrificing their invasive and migratory properties and potentially regaining some of the proliferative abilities which are thought to be lost during EMT. So this is the paradigm which has been in the field for quite some time, and that these cycles of EMT and MET continue to form metastasis throughout. So if you look at the field in the sort of first decade of this millennium, the century, this has been viewed as a very binary process where cells are either epithelial or more mesenchymal. The third possibility where potentially cells can migrate collectively, or that cells can have a given phenotype which is not necessarily completely epithelial or completely mesenchymal, was not really very well mentioned in the literature. Wherever it was thought of as to be a metastable or a transiently stable state, but not really a state in which cells can actually perform the entire process of metastasis. What has happened over the past decade is that this thought process has undergone a major change, and now people think that there are multiple states of EMT, starting from being very epithelial to being very mesenchymal, and these states, these hybrid states, one or more which may exist, they actually have properties such as higher metastasis as compared to those which are completely epithelial or completely mesenchymal. So not only these hybrid states exist, but these hybrid states may actually be the most aggressive ones. This is some of the initial mathematical modeling that we had done, and some other people sort of continue to develop models, which I'll go into in a bit. So as I mentioned, these cells are thought to be potentially more fit for metastasis as compared to those which are completely epithelial and mesenchymal. This is a snapshot from Nature paper where they talked about in vivo squamous cell carcinoma models and showed that there were multiple states of EMT. Cells could switch back and forth because, as you saw on the previous slide, that there is a possibility of switching back and forth depending on various intracellular as well as extracellular signals that cells might be receiving. And the ones which are most metastatic are not the ones which are completely epithelial or completely mesenchymal, but the ones which are somewhere in between. So what we did here was we had earlier developed this EMT scoring method where if you give a microarray or an RNA-seq profile, it can quantify the extent to which this particular sample has undergone an EMT. This is a statistical method based on multinomial logistic regression where we calculate the probability of a sample belonging to either epithelial, mesenchymal, or hybrid, and based on those probabilities, you can bin them into one category, and the score that you get is on a spectrum of 0 to 2. 0 means fully epithelial, and 2 means fully mesenchymal. There have been two other methods which have come up in the field, which use slightly different gene lists and different algorithms. So we first wanted to see that is there any consistency in EMT nests calculated by these three different methods, and do we see some interesting trends with respect to the hybrid phenotype? So what we did is that we looked at the CCLE Cancer Cell Line Encyclopedia, which is a compendium of cell lines, more than 900 cell lines across different tumor types. And for every tumor type, which is shown by a different symbol here, we calculate, there were multiple cell lines, so we calculated the average as well as the variability, the variance in this case, using all the three methods. Interestingly, what we saw was that the ones which were most variable actually showed out to be the common across all the three methods, suggesting that irrespective of which of these three gene lists and algorithms that you were using, some common trends showed up, which were that the more the number of hybrid samples that were present in any given tumor type, the higher was the variability there, suggesting that the presence of hybrid is actually contributing to higher heterogeneity in that context. And we found across multiple different data sets that we have looked at, these three methods correlated quite well with one another in terms of pairwise correlations, again suggesting that there is potentially some overall global EMT signature which can be captured by any of these three methods. We next wanted to see if this property of hybrid can actually be used to identify certain more aggressive cancers. In this context, we looked at inflammatory breast cancer, which is highly metastatic. It has a 5-year survival rate of less than 30%, and 1/3 patients already have metastasis at the time of diagnosis per se. It does not present as a dominant mass, so that's even more difficult for clinicians to identify it. There's no molecular therapy that is out there yet, and interestingly, no unique mutational signature or even gene list that has yet been identified for IBC or inflammatory breast cancers versus non-IBC. What we definitely know is that this metastasis is largely via collective behavior, and using previous observations that our collaborators and others have made, it looks like that this might be a hybrid phenotype. So our hypothesis was that if that is true, then potentially one way to identify IBC can be that there is more heterogeneity in EMT-ness when it comes to IBC as compared to non-IBC. So we looked at all the different gene lists that have been so far identified, that have been so far proposed to identify IBC. And we first wanted to see if there is any overlap of genes between those. And to our surprise, we found that basically these 4 gene lists were completely disjoint. There was only one exception, that 2 of these gene lists had one gene in common, but everything else was completely different, which we took us by quite a bit of surprise, that if all of these gene lists are claiming to do the same thing, then potentially you would expect that there are some common conserved molecular processes and or genes which did not just show up. Then we looked at 3 other publicly available data sets, and for which we already know based on pathological validation which of the sample is IBC versus non-IBC, and we calculated the mean and the coefficient of variance of the EMT scores using each of these 3 methods in each of these 3 data sets. What we found was that in 8 out of 9 cases, the variance in IBC was more than the variance in non-IBC, suggesting that There may not be a unique gene signature, but one clear thing that shows out to identify IBC is that there is more heterogeneity in terms of EMT. So that heterogeneity itself can be a signature, which has the potential of being quantified through these methods that we have been looking into. So this idea of phenotypic plasticity and heterogeneity, as I said, is not something which is just even in cancer. It's there in developmental biology. It's also there in evolution of different species. There was this recent book that I was involved in co-editing where we had collated evidence across different organisms, across evolutionary developmental biology contexts, and across different diseases, including cancer, where this idea of phenotypic switching, plasticity, heterogeneity, their ability to go back and forth, adapt, again, not at a genetic level, but at a phenotypic level, has been investigated in quite some detail. So one of the themes that shows up, which again takes us back to the example that I was talking about, is the ability to have multiple states, which is the property that is called as multi-stability, that if there is a system in which there are multiple states, and states here being defined in this representation of Addington landscape, and if you were to sort of go back to your earlier high school physics, a stable state is defined as that if you give this cell a perturbation or this ball a perturbation, if it comes back, then that's a stable state, and if it disappears or it goes away completely, then that's an unstable state. So in this representation, a valley would be a stable state, and this sort of mountain-like structure would be an unstable state. Now, what are the implications of having these states? So if you have two states, given there is enough noise in biological systems, noise here meaning uncertainty in various different biochemical reactions that might be simultaneously ongoing within a cell, it is possible that cells can switch their states. So for instance, again, this is just a slightly more physicsy representation of the Addington landscape, where what you see is that if you have these two states, then how you would realize that in a flow cytometer is basically by having these bimodal distribution. You have two peaks. Now, if you were to sort out, just segregate these cells, and what is expected to happen is that if you only start with the X state cells, so first day, of course, that is what you are, but later what you see is that this LX was able to give rise to this HX population. So without any induction being given, these cells can switch back. Now, this has important implications in terms of therapies, because one might think that one has deleted or deprived or killed all the sensitive cells. But even if the resistant cells are remaining and the resistance is emerging due to multistability, these resistant cells can give rise to sensitive cells, or even if there are a few sensitive cells remaining, those sensitives can reversibly switch to a resistant state. So of course, there are important implications of this particular process in the context of cancer progression and so on. So the questions that we started asking was that now if you look at different networks that regulate these different processes, do they have this property of multistability? Can they give rise to two or more states which would allow switching back and forth in a reversible stochastic manner? So this is the network that we started looking at in the context of EMT this couple of years ago. It's a very small network, very simple network, has only 4 players. Of course, EMT is much more complicated than that, but we just wanted to start somewhere. So here are 2 transcription factors that drive a more mesenchymal phenotype. There are 2 microRNAs which drive a more epithelial phenotype. And what you see between them is a toggle switch, or is a mutual inhibition, some sort of a tug of war, which makes sure that cells are either in epithelial or in mesenchymal. We had to develop mathematical modeling framework for it. I'll not go into those details, but I'll just highlight the major predictions that we made at that point of time. So we made these predictions that, first of all, EMT is not a binary process. Cells can have this third stable state. The other predictions we made, so here each of the blue lines is a state. The x-axis is EMT induction signal. The y-axis is a measure of EMT-ness in some manner. What you see is that there are regions in which there is more than one blue curve, suggesting that if snail levels are somewhere here, it is possible that cells can go back and forth. So suggesting, again, that if you take an isogenic population, which has the same genes, all cells have the same genomic background, but still you can have phenotypic diversity. So the genotype to phenotype mapping is not 1-to-1 per se. These predictions have then been experimentally validated by various of our colleagues and others independently. this is a lung cancer cell line showing stained with green using ecadherin, which is an epithelial marker, and red is vimentin, which is a mesenchymal marker. And what you see is that many cells actually co-express both of them, suggesting, and they continue to do that over multiple passages, suggesting that this might be actually a stable hybrid phenotype. If you do a flow cytometry of the same proteins that I just talked to you about, using multiple different cell lines belonging to breast cancer, prostate cancer, lung cancer, and colorectal cancer, you still see this heterogeneity that persists. And interestingly, what we also found, and this was the most interesting experiment to us, where these cells were segregated based on EpCAM, which is again another epithelial marker, and vimentin-associated GFP, which is the same as the y-axis here. We saw that there were these three states in this prostate cancer-derived cell line from a mouse model. And now, if you segregate these cells, you see that these cells are able to give rise to others. And this is the exact same thing that I was talking about a couple of slides earlier. If you isolate only one cell population, you can actually get back the other population without necessarily having any induction. Those are the important aspects of this. What has happened since is that various different people have started modeling EMT networks. They have developed models for larger and larger networks as experimental data for EMT has been growing using different modeling strategies and so on. It's a very active field. What I'll just briefly talk about is some work that was led by my PhD student and two summer interns together with our collaborators in Italy. So what we did was we looked at multiple different networks here that people have been curating and characterizing, including us, of what regulates EMT and MET. And we simulated them using different strategies. And the question we were asking is that, is there something fundamental about the way these things are connected that allows these features? So basically, if you were to, say, switch these connections, so instead of ZEB self-activation, if, say, ZEB self-inhibits, and instead of GRHL2 inhibiting ZEB, if GRHL2 activates ZEB. So if you switch these two connections, what happens to the properties of this system? Why are systems designed the way they are is the question that we are asking. So again, we were taking a very simple physics approach, very common in physics kind of approaches that, you take a simple system, there is a ball rolling down a pulley, there is no friction, etc., etc. You write down a set of equation and you can actually very well characterize what the behavior of this ball rolling down this inclined plane would be. So a very simple approach. What we saw, when we simulated this system, what we observed was that this system can give rise to two states. So it has the possibility of giving rise to a mesenchymal state, which has very low levels of ecadherin, and epithelial state, which has high levels of ecadherin. We looked at larger networks. What we found was, of course, there were these two states, which were still most dominant. These are just cartoon representations of detailed quantitative analysis that we have done. And you begin to see many, many more states. The larger the network you take, you see more states. And you see one constant feature was that the epithelial and mesenchymal were still the most dominant states per se. Then we did these in silico experiments, because you can do these sort of experiments on computers, where what you do is that for every network simulated, we either deleted an edge, changed the sign of an edge from negative to positive or positive to negative, or added a hypothetical edge where one did not exist. And we saw different kinds of results. So we saw, so this is what we call mutant type 1, type 3. So what we observed was that, there was one most dominant set of cases where this distribution changed a little bit, but there was no qualitative difference. Then there were other kinds of cases where the cells were basically depleted of the ability to give rise to other cell types. So here, what you have done is that you have reduced heterogeneity because you have gotten rid of the other state. It can be epithelial or it can be mesenchymal. So what was happening in this case is that if we were disrupting feedback loops, which were positive in nature, then such possibility was coming down. So what we saw was that the higher the number of positive feedback loops in a circuit, the higher the heterogeneity or the plasticity and so on. So we suggested that if you break feedback loops, then that can be one way to reduce heterogeneity into the system. And this is what was exactly done in a paper recently, where they had looked at the same network that I showed you earlier, and ZEB inhibits MER200, MER200 inhibits ZEB. So overall, this is a positive feedback loop, negative multiplied by negative. So this was giving rise to some bimodal distributions, as I've been showing earlier, and then they deleted this using CRISPR. What they saw was that in the mutant cell line where this feedback loop was broken, metastasis was significantly reduced. Now, this paper does not go too much into details of why this happened, but I think this gels very well with some of the predictions that we have been making and the way we have been interpreting data, is that, of course, you have decreased plasticity, you have less chances, you have less heterogeneity. If there are cells of different types, then it is possible that they may be cooperating. So there can be both cell autonomous as well as non-cell autonomous contributions to this, and both of which can be decreased in some way. Now, this thing was something which, as I mentioned, was very intriguing to us, that when you segregate these cells, this experiment that I earlier also highlighted, then the epithelial and mesenchymal largely maintain themselves. So if you look at their percentage over a period of 2 weeks, they are somewhere 70 to 80% maintaining themselves. But the hybrid one quickly gave rise to other cell types. So we wanted to ask that, can we explain these features of population dynamics? This work was led by Shubham in Herbert Levine's group, which also happens to be my previous supervisor at Northeastern. And we asked this question, that since these experiments are at a time scale of multiple days where cell division is also happening, we of course need to incorporate cell division. So that the next important question becomes that, do we know what happens to the EMT status of a cell when a cell divides? Does it remain the same? Does it always change? Or again, is there an element of stochasticity or probability? So there's no experimental information that is available to us so far that we were able to find. So the assumption that we made is that every time a cell divides after whatever 30 hours or so, there is some probability of asymmetric cell division, that some molecules might be distributed with some noise. So if, say, there are 10 molecules of ZEB1 in the mother cell, not every daughter cell will get 10 and 10 after the DNA is duplicated and so on. They might get 4 and 6. So we included 10% noise. So you can't go from 0 to 10. The noise is only 10% of the distribution. And ask the question that, if you take those assumptions, can you reproduce this experimental data? Can our model explain this data? And largely speaking, yes, we were able to see that here the X and Y axis are the same as you saw on the experimental data. The dotted line is experimental data, the solid line is simulation data. We saw that just making this simple assumption for a simple 4-node network of EMT that I showed you earlier, we were able to explain many features of this experimental data, suggesting that yes, it is possible that cells switch their EMT phenotype during cell division as well. And that's a prediction that still remains to be experimentally tested. But what is the possibility? If this is true, then what are the possibilities that might be happening? So if, again, I will request you to go back to sort of high school physics, where once you learned about magnetism, so if you have an unmagnetized substance and then you expose it to increasing dose of magnetic field, this all the sort of micromagnets in that get magnetized in a specific direction, which is the same as the direction of the magnetic field. Now, if you withdraw the magnetic field, you do not see that when there is zero magnetic field, there is zero magnetization. No, that does not happen. There is some memory that is built up into the system, and you have to apply the magnetic field in the opposite direction to make the system go completely unmagnetized. And this is what a property is called hysteresis, which means that the direction, the path that cells take from A to B is not the same path as what they take from B to A. So asymmetry in paths is what is called as hysteresis. So we were actually able to see this in our simulations, that if you induce EMT for 10 days and if you withdraw the signal for 10 days, the distribution is not the same. When you withdraw the signal, you still have around 40% of the cells that are still mesenchymal. So this can be another effect in this particular case, and there has been experimental data more recently that actually shows this is what is happening. Just the last part, we also saw that since this distribution can continuously change, what can happen is that what distribution that you see in, say, a cell line today might not be the same as distribution that we see later. And this is exactly what we saw. If you take multiple different data sets from which you take these, say, A549 or others, you actually see that their EMT score is quite varying. So again, suggesting that the EMT score of a cell population today and later might also be different, because this is a dynamic property and cells are continuously changing. And that's the idea of non-genetic heterogeneity. So I would just end with the thought that, clearly we have been thinking about cancer as a genetic disease, but that's not about it. There are various other components that can be studied using these quantitative approaches, and of course, while being in a constant collaboration with the experimental data and clinicians. And the questions that we sort of begin to focus on: where does heterogeneity come from, the role of this heterogeneity, and can we control this? We have some answers to that we have begin to gather in the first aspects that I touched upon, the multistability and noise. And we are still working on the role of this heterogeneity in terms of developing models to actually show that heterogeneity leads to higher fitness, and so on. And I would just end with these last sentences of the most famous reviews that have been written in the field of cancer biology, called Hallmarks of Cancer. So the first review, written in 2000, ends by saying that one day we imagine that cancer biology and treatment will become a science that has a logical coherence that rivals that of chemistry or physics. Then these, I'm sure you're aware, are disciplines where mathematical predictions are being made, and then they're tested by the community, and this iterative process continues, something which is much less commonly seen in biology, and specifically in cancer biology. And 10 years later, they wrote that we still do not completely understand these underlying organizing principles. We know there is complexity, but potentially there are some hidden laws which we still need to figure out. With that, I would like to thank the fantastic set of people I've had the fortune to work with, who come across different disciplines, because clearly cancer is a disease that we need disciplines and insight. We need insights coming from all different disciplines. And I would thank once again Zinia for the kind invitation, and the organizers of SCS 2020, and I'll be happy to take any questions.
Thank you, Mohit, for a very interesting talk. Colleagues, maybe you have any questions? Oh, yeah, we have one question in chat from Nikita Novikov. May I read this question, Mohit?
Yes, please.
It's known that p53 gain of function mutation induce EMT in various cancers. Will this p53 gain of function mutation fixate cancer cells in certain phenotype, for example, in mesenchymal phenotype, or will cancer cells still be able to switch states? Thank you for the interesting talk.
Thank you. I think that's a very important question, because clearly the genetic background does affect the EMT status. So p53 gain of mutations would make the cells more likely to be in a mesenchymal phenotype. The reversibility of this process is also will be dependent on the other mutations that cells already have. So if they have a mutation that can drive the cells to a more epithelial phenotype, then there will be a competition there. So alone p53 mutation might make cells relatively strongly mesenchymal, and potentially an irreversible transition also. But the combinatorial effect of other mutations will be important in answering these questions in a quantitative manner.
We have the reply from Nikita. Thank you, Mohit. Mohit, I have one more question about EMP states in circulating tumor cells.
Yes.
Do you know this information, and how different EMP states can be relevant, can be important for CTC survival, for example?
Yes, I think that's a very important question. Thanks. So we have looked at some CTC data sets, and we do see the heterogeneity there also. We have come across very few data sets which have both primary tumor and matched CTC. So it's hard to say whether one can predict the EMP-ness of CTCs based on EMP-ness of primary tumor or vice versa. But based on some clinical studies that I've been coming across, the suggestion is that a higher enrichment of these hybrid cells in CTCs is also associated with a worse prognosis in multiple cancers.
Okay, thank you. And another question is regarding inflammatory breast cancer.
Yes.
You showed extremely diverse number of EMP variations, variability in this type of breast cancer, but what is the potential mechanisms of this variability? Because only one gene, only one protein, E-cadherin loss results in the formation of this breast cancer.
So we are still waiting for any publicly available single-cell RNA-seq data in IBC to be able to address these questions. So far, whatever I presented is at a bulk level, and these, I mean, here different samples are different patients per se. So we want to see if the patient's sort of inter-tumor heterogeneity maps onto intra-tumor heterogeneity. That still remains to be seen because we just don't have that data. Once we have that data, then we can begin to answer more questions. Because currently, because it's inter-tumor, it will also be affected by the genetic background. So I cannot claim it to be isogenic cells.
And thank you, Mohit. And another question in our chat from Marina Patishova. How does an inflammatory type of breast cancer intersect with a molecular subtype? Is it a triple negative subtype?
So inflammatory breast cancers have been seen across different subtypes. There have been some reports suggesting some enrichment of triple negative, but no exclusive data that it's only triple negative. Other subtypes have also been witnessed.
Okay, thank you very much, Mohit, for your interesting talk, for your time to present your talk in this difficult time. And now we have a break before 16, and we will see after 1 hour. У нас сейчас перерыв 1 час, и мы увидимся с вами в 4 часа.
Thank you so much, Senior. Thank you.
Thank you.
Bye.